Jai Gurudev students. Our next part is cell inclusion. Now these cell inclusions are non-living structure of the cytoplasm and are also called deutoplasmic or ergastric bodies. Now about the vacuoles. The occurrence of the vacuoles are found in all the eukaryotic cell type. In animal cell, their number is small in size, but number is more and structure is small in size and irregular, scattered within the cytoplasm. While in the plant cell, there is a single large central or sap vacuole filled with a cell sap. It occupies about 90% of the cell volume. Now, in case of the structure is form of two parts, tonoplast and cell sap. Now what is tonoplast? It is the outer single layer, an outer membrane and semi-permeable in nature. It transports ions and other material by active transport system. And next one is cell sap, and this cell sap is liquid present inside the central vacuole and is formed of a water, mineral, sugar, amino acid, protein, alkaloids, etc. And what is the function? Function is cell sap extract turgor pressure, means pressure towards the wall due to the fully flaccid condition. It stores water, nutrients and mineral. Sometimes it is also store waste. And the vacuoles are modified in the different forms. Some vacuoles are food vacuoles, and some vacuoles are known as contractile vacuoles, and some vacuoles are known as the pseudo vacuoles. That is, in case of the food vacuoles, they contain the egestate or the digested or undigested food. And next one is contractile vacuoles. These are found in the freshwater protozoans, just like amoeba, and help in the osmoregulation. Gas or air vacuoles, or you can say the pseudovacuoles, these are found in the cyanobacteria, that is blue-green algae, and photosynthetic bacteria, and provide buoyancy or upward thrust. Okay. Next one is reserve food. This reserve food, it may be in the form of a starch grain or the glycogen granules or the protein granules or the oil droplets. And the categories are gaseous. It includes oxygen, carbon dioxide, nitrogen, waste, ammonia, urea or uric acid, inorganic crystals, sometimes it is present, crystoliths as a crystoliths, okay, and raphides, and some of this reserve food as a secretion found in the granular cell and included mucus or resin and gum. Our next one is the most important one, that is the nucleus, which is very important one, and it was first discovered or described by the Robert Brown in 1831. And definition, the nucleus, it's also called the director of the cell, and it is the most important part of the cell which direct and controls all the cellular function or the activities. In the true nucleus, that is, in a true nucleus is present in all the eukaryotes except mature mammalian RBC, sieve tube cells of phloem and tracheids and vessels of xylem. Now position, generally they are centric, but sometimes it is peripheral in nature, and number, mostly the cells are monokaryotic, but sometimes maybe two nucleus or more nucleus may be present. Okay, and its shape are of different shape, different types. Some are that is round in shape, oval in shape, bean shape in shape, horseshoe shape, and elongated shape. Two lobules or more than two lobules may be present. So these are of different shape. Now the most important one is ultrastructure of the nucleus. You just see the structure of the nucleus. It is bilayer, two layer is present. In between then that is pores are present, which is known as nuclear pore, and this two bilayer is lipoproteinous in nature. Outer membrane with the ribosomes, inner membranes with fibrous lamella which are called honeycomb layer. You just see the outer layer with the ribosomes and inner layer with a honeycomb, that is comb like structure, honeybee's comb like structure, okay, and fibrous lamella. Two membranes are separated by means of a fluid filled perinuclear space, and that one is this membrane is porous, and all of you know this membrane is porous. Pores are filled with a cylindrical structure which is known as annulus. This pore, within this pore, this structure is present which is known as pore complex, and this sharp structure, total structure is known as annulus. Now this nuclear membrane, they have a chromatin threads, red color. So chromatin threads are present, and this is known as endosomes or the plasmosomes, these structures, and this one is known as nucleolus. This is nucleolus. This is known as chromocentry. This structure is known as chromocentry. So this is the structure of the nucleus. Now what is the function? It allows the passage of inorganic ions through this gap, and it helps in pinocytosis or phagocytosis, the terms you know, and fibrous lamellar help in dissolution and reformation of nuclear membrane. Then nucleoplasm which is present, it is transparent within the nucleus, that is plasma or the fluid structure. It is transparent, homogeneous, semi-fluid, colloidal and ground substance present inside the nuclear membrane. And the function of this, that is nucleoplasm, is it acts as a nuclear skeleton and helps in maintaining the shape. It helps in the formation of the spindle of protein, synthesis of ATP, NAD, RNS. Okay, so this is the function of nucleoplasm. Our next one is nucleolus, and this nucleolus is observed by the Fontana in 1781, and position it is associated with a nuclear organizer region of chromosomes, and it is absent in muscles, fibers, RBC, yeast, sperms and prokaryotes. It is a naked with no limiting membrane. Central part is called fibrillar zone. Okay, central part is called fibrillar zone. Fibrils and granules collectively forms the nucleonema. Outer nuclear associates, that's perinuclear chromatin and intranuclear chromatin. Perinuclear outer and inner, intranuclear. Matrix, these are the matrix. Some granules are present, and within that granules, in between that granules, fibrils are present. Okay, so this is the structure of nucleolus, and the chemical composition, it contains rRNA, and function, it is a set of biosynthesis of rRNA and stores rRNA. It helps in the spindle formation and helps in the transport of rRNA, messenger RNA and tRNA. Okay, the next one is nuclear chromatin. It is darkly stained network of a long and fine thread called the chromatin fibers. There is a two chromatin fibers, one is heterochromatin, another one is euchromatin. Sorry for the sound. And heterochromatin, it is formed of a thick region with a condensed DNA, and euchromatin, that's euchromatin contain fibers are chemically formed of a DNA, RNA and protein. During cell division, these chromatin fibers condense into number of rods called chromosome. Now, let me describe the structure of the chromosome. Chromosome consists of a two identical and spirally coiled thread, and which is called sister chromatids, and joined at lightly colored constriction, and this and called the centromeres. Okay. So see the structure. So here is a two, they are coiled with each other. Chromosome 2 are, that is two, here you can see the two, and here also the two chromatids. Some constriction, this constriction is known as secondary constriction, and it is known as primary constriction, and just a bulging part which is known as satellite. So in middle, primary constriction is present, secondary constriction, these two are the secondary constriction, and above the secondary constriction one satellite, that is known as satellite, and that is known as pellicle, and this primary constriction, and this one is known as kinetochore. Okay, and these two chromonymeta is called chromonymeta. Okay, this is called chromonymeta. Now the position that is on the basis of the number of the centromeres, the chromosome R is called monocentric or dicentric, and on the basis of the position chromosome are two types, that is centromeres, on the position of the centromeres, and how it is structured. If centromere is terminal, then telocentric. If the two arms are not unequal, acrocentric, and if the two arm is slightly unequal, submetacentric, and if two arms are equal, this is known as metacentric. So this fourth type is very, very important for you. Okay. In the electron microscope, the structure shows that the chromatin fiber is formed of a chain of repeated units called nucleosomes. And what is this nucleosomes? Each nucleosome is formed of a core complex. This one is known as core complex. Okay. This core complex is formed of a histone octomere. So that is 4 type of histone. 2 molecule each, that is H2A, H2A, H3, H3, H4, H4. Okay, this H2B, in another side, another H2B is present, but the difference is that this H2, that is the DNA segments between the adjacent nucleus is called the linked DNA. So this one is linked, this histone, linked DNA, and it helps in grouping the nucleosomes, not pass to the next generation. This H1 is a single one. This H1 helps in grouping of chromosome, just to group down the chromosomes, and it will not pass to the next generation. Rest of the all that will pass to the next generation. Okay, now it is clear.